So in this video, we're going to discuss some components of the cell membrane and how they work. So this is a diagram showing the cell membrane. None of these components should be surprising to you. You can see this is the inside of the cell where the cytoskeleton is, and out here is the outside of the cell with the extracellular matrix. You'll notice that here's our lipid bilayer membrane, and there are quite a few molecules actually inserted or associated with the membrane. So these molecules that go all the way through the membrane, these happen to be proteins. These are called integral membrane proteins. Molecules such as proteins that are only on one side or the other of the membrane are referred to as peripheral proteins. You'll also notice that there are quite a few glycosylations, meaning that there are carbohydrate molecules attached to many of the proteins and lipids that are in the membrane. These carbohydrates are very important for cell-cell recognition and in differentiating different species or different types of cells within an individual. So the membrane has a very important function. This is a fundamental function of being alive, maintaining a difference between the inside and the outside environment. So let's talk about how that works. So first we need to know a little about what the membrane is made of. What are the components of it? We know a little about this already. These are our phospholipid membrane molecules, and this again is a bilayer, so you've got one layer pointing this way and the other layer pointing the other way. Here's our phosphate head groups, and each of these has two carbohydrate tails associated with it. So these head groups are hydrophilic. They associate well with water. The tails are hydrophobic. They do not allow water to pass through easily. And that's really what maintains the structure of the membrane. So the membrane we saw has quite a few molecules inserted into it. This is an integral membrane protein, and there are some important aspects to this protein. So one important thing is that the edges of the protein, the ones that stick out to the extracellular or the cytoplasmic side, are hydrophilic. They associate well with water, just like the phosphate groups on our lipids do. The center of this protein is going to be hydrophobic, and that's really what keeps it anchored in the right spot in the membrane. So proteins in the membrane have lots of different functions, and we'll mention several, but these are not the only ones. So first of all, many proteins in the membrane are involved in transport processes, and we'll talk about those in detail in the next video. Many proteins have enzymatic functions. Some of these are signaling related, and some of them have other enzymatic functions. Some proteins are involved in attaching the cell to the extracellular matrix, keeping it in the right spot, or attaching particular molecules in the membrane to the cytoskeleton. Some proteins are involved in cell-cell recognition, again, often involving those carbohydrate molecules. Some proteins, are involved in very important tight interactions between certain types of cells, and we'll talk about those more in detail much later in the year. A final function, not the only functions again, but one other important function is signal transduction, and we'll talk about that in detail in the third video of this unit. So now that we know a little bit about the components of the membrane, we need to know a little bit about how it works and how it behaves. So one thing that we know about the membrane is that it's not a completely solid structure. The lipids themselves and the proteins within the membrane move around quite a bit. And one of the experiments that showed that this is possible is a simple experiment where they literally fused a mouse cell with identifiably mouse proteins and a human cell with identifiably human proteins. You fuse those two cells together, you wait a little while, and you come back. And what you see is that instead of having a cell with all the mouse proteins on one side and all the human proteins on the other, you actually see that those proteins have mixed together quite effectively. Sometimes this mixing is random and sometimes it's actively controlled by the cell. Molecules can be moved within the membrane by the cell, but certainly the molecules in the membrane can move around quite a bit. So what makes the membrane so fluid? There are a couple of different aspects of the membrane or characteristics that control how easy it is for molecules to move within the membrane. So one thing is the composition of the phospholipids themselves. So phospholipids that are unsaturated tend not to pack very well together. And we know this because unsaturated tails tend to have these kinks in them, whereas saturated tails pack quite tightly together. And these types of membranes tend to be much thicker, much less fluid than membranes with a lot of unsaturated tails in them. So that's one thing that can vary between membranes and control the level of movement of molecules within the membrane. Another important aspect is what exactly the membrane is made of. So obviously the main part of the membrane is these phospholipid molecules, but in many cases there are also additional molecules inserted into the membrane that help regulate how fluid that membrane is. And one of those molecules is cholesterol. Cholesterol tends to reduce membrane fluidity at moderate temperatures, and you can imagine that these cholesterol molecules are sort of hard to move around. But at low temperatures, the presence of this cholesterol can prevent the membrane from solidifying and essentially freezing. These kinds of differences are very important evolutionarily. You can imagine that organisms that live a lot in cold environments or have seasonal periods where it gets very cold might have quite a bit of cholesterol in their membranes. Organisms that live at very high temperatures might have much more viscous, saturated phospholipids in their membranes. Just a reminder about how these proteins get into the membrane. Remember, here we are in the endoplasmic reticulum where the proteins are being made. Those molecules then move out to the Golgi. The proteins are actually, membrane proteins are inserted into the membrane as they're made and as they're folded. Many of the glycosylations, the additions, happen in the Golgi apparatus, and then those molecules eventually make their way through vesicles from the Golgi out to the plasma membrane or potentially to other membrane-bound organelles in the cell. And that is an introduction to membranes.